Can DNA demand a verdict? In 1987, forensic DNA analysis made its first appearance in a U.S. courtroom. Originally known as DNA fingerprinting, this type of analysis is now called DNA profiling or DNA testing to distinguish it from traditional skin fingerprinting. Even though it is used in less than 1% of all criminal cases, DNA profiling has helped to acquit or convict suspects in many of the most violent crimes, including rape and murder. How can DNA be used to identify an individual? Almost every cell in our bodies contains DNA, which is the genetic material that programs how cells work. Any two people share, on average, 99.9% of their DNA, meaning that only 0.1% of your DNA is unique to you. The only exception is identical twins who share 100% of their DNA. Each human cell contains 3 billion DNA base pairs. Our unique DNA, which is about 0.1% of that 3 billion pairs, amounts to 3 million base pairs. That's more than enough to provide a profile that accurately identifies a person. DNA is often left behind at a crime scene. It is present in all kinds of evidence, including blood, hair, skin, saliva, and semen. Scientists can analyze the DNA and evidence samples to see if it matches a suspect's DNA. Here, we can see how DNA evidence used to be collected and analyzed. In its early days, DNA analysis required an evidence sample of at least the size of a dime. Today's techniques can amplify the DNA using PCR or polymerase chain reaction, which makes millions of copies from the tiniest amounts of evidence. This approach is also helpful for analyzing poor quality DNA and evidence samples collected from dirty crime scenes or in very old samples. Blood relatives have more similar DNA. Unrelated individuals have 3 million DNA bases that differ between them, but blood relatives have fewer differences. If the DNA profile from a piece of evidence is similar but not identical to that of a suspect, officials may investigate blood relatives of the suspect. What about human error? Forensic investigators take many precautions to prevent mistakes, but human error can never be fully eliminated. The National Research Council, or NRC, recommends that evidence samples be divided soon after they're collected so that if a mix-up were to occur, there would be backup samples to analyze. To detect possible contamination of DNA samples during collection or handling, DNA profiles are typically compared with those from the detectives at the crime scene, the victim, a randomly chosen person, or a DNA profile from a database. Each sample is assigned a code and testers don't know who the sample came from. The NRC recommends that forensic DNA analysis be conducted by an unbiased outside laboratory that maintains a high level of quality control and a low error rate. Is DNA evidence alone enough to acquit or convict? It is easier to exclude a suspect than to convict someone based on a DNA match. The FBI estimates that 1/3 of initial rape suspects are excluded because DNA samples fail to match. Forensic DNA is just one of many types of evidence. Investigators also look at other clues such as motive, weapons, alibis, and additional evidence linking a suspect to the crime scene. When multiple lines of evidence tell a consistent story, investigators can be assured that the samples from a particular suspect were not planted, either on purpose or by accident, at the crime scene. Can DNA evidence exonerate wrongfully convicted prisoners? The Innocence Project at New York's Benjamin N. Cardozo School of Law aims to exonerate prisoners wrongfully convicted of crimes. The project uses DNA profiling evidence to support the reevaluation of criminal cases. But DNA evidence alone is not enough to get a person out of jail. The case must also be re-examined by a judge, along with lawyers representing both sides of the case. Since 1992, the Innocence Project and others have used DNA evidence to exonerate over 300 prisoners, including 18 who are on death row, one of whom was only 5 days from execution. While some states will reconsider cases with compelling DNA evidence, regardless of when the trial ended, several states restrict the time for post-trial submission of DNA evidence to 6 months or less. And unfortunately, the evidence from some cases have been lost or destroyed, making DNA analysis impossible. The Innocence Project seeks to reform the criminal justice system in ways that will improve access to DNA testing, improve evidence preservation, and permit reevaluation of DNA evidence in closed cases. Is DNA profiling technology used appropriately for justice? DNA profiling can be a powerful tool in criminal investigations. Its success in the courtroom depends on many factors, including proper handling of evidence, careful analysis by an unbiased forensic laboratory, fair and appropriate interpretation of the results, and accurate and effective reporting of the results to judges and jurors. When used correctly, DNA profiling is a powerful forensic tool. It can be used to quickly eliminate suspects, saving time in searches for perpetrators. It can also provide compelling evidence to support a conviction and, most importantly, to reduce the chances of wrongful conviction. Restriction fragment length polymorphisms, or RFLP, as shown here, is an older DNA profiling technique. DNA evidence is collected from the crime scene. Then, the DNA is isolated from the evidence sample. The DNA is cut into fragments using a specialized protein called restriction enzymes. For every person, the size of cut fragments are unique, except for identical twins. We can separate these fragments by passing them through a gel in gel electrophoresis. Then, we can transfer the DNA fragments from the gel to a sheet of membrane and probe the membrane with DNA fragments that complement the DNA sequence of the fragments of interest. Then, we can compare the fragment profile of evidence DNA with those of suspects, as well as people we should rule out, like the detective and the victim, to see if they match. We can reprobe the membrane up to 10 more times to identify different fragments. If the profiles from the evidence DNA and the suspect match multiple times, then it is very likely that the evidence DNA came from the suspect. Can you tell by looking at this gel which suspect matches the evidence DNA found at the crime scene? Thank you for watching.